So welcome to Lab 1. This is the eyeball. So if you don't have your checklist out, please get it. But let's look at the checklist real quick. So remember, a checklist is exactly what it sounds like. It's a list of the structures that we're going to learn for a particular system or particular organ or whatever. Well, if you look at this one, it's more than just the eyeball. This is everything for the eye, the ear, and the endocrines. So Emmy asked, was that first collab graded or just, none of the collabs are graded, Emmy. Your grades just come from quizzes and the lab and lecture exams. So no, it wasn't graded. It's not mandatory that you attend any collab except that one was just to get the class started. The rest of these are just going to be for lab or for a review. But anyway, let's continue. So is it just for the eyeball? It's all the eye, the ear, and the endocrine system as well. But today we're just going to be looking at the eye. And so for most books, this is page 364. And then these are the structures that you're going to have to look at. So let's go ahead and do that. So first of all, we're going to be looking at the eyeball itself. But look, this says eyeball with lacrimal apparatus. The word lacrimal just means tears. Lacrimate means to make tears. And so it's also going to be the part of the eye that has to do with tear production. And so when you look at it, these are some of the models that if you were in the face-to-face that we would use. So look, this is a model of just the eyeball. This model has not only the eyeball, but it also has this lacrimal apparatus around it. And then here's a coronal section through the eyeball so that we can look on the inside. So these are the models that we're going to look at. And this is one we're going to look first. And so if you have your checklist, we're going to look at it. And then we're going to look at this one as well and this one. So the first thing we're going to look at, though, are muscles that move the eyeball around. So there are actually 6 muscles that move the eyeball around. They're all skeletal muscles, which means you have voluntary control over them. But there's 6 of them. And so humans can move their eyeballs in lots and lots of different directions. If you were taking this on campus, we would do a cow eye dissection. But if you look at the cow eye, it doesn't have 6 muscles. It only has 4. And so cows can't move their eyes in the same directions that humans can. They can move their eyes up, and they can move their eyes down. They can move their eyes left, and they can move their eyes right. But that's it, up, down, left, right. Humans, we have 2 extra muscles. So not only can we move our eyeballs up and down and left and right, we can also move them diagonally. So we can move them this direction and this direction. And so it makes our eyeballs very, very mobile. But let's look at these muscles. So when we look at them, every single one of them is red. Red usually designates skeletal muscles, and that's what they are. So there are 4 muscles that are straight. And the word for straight is rectus. So we're going to see the rectus muscles. Well, they're named for the positions on the eyeball. And so we have to know the position of the eyeball. We have to know superior, and we have to know inferior, and we have to know medial, and we have to know lateral in order to name these, because that's what they're named. And so there has to be some sort of landmark to let us know these things. And that landmark is this. So this is the lacrimal gland. And the lacrimal gland is always on the lateral side. So it's on the side of your head where your temples are. It's on that side. So if this is lateral, that means this is lateral. That means this is medial, and then this is superior, and this is inferior. And that's how we're going to name those. So there's that lateral gland, the lacrimal gland. So here's the lateral rectus muscle. Rectus means straight. So when you look at it, it's a straight muscle. And so the opposite side is going to be the medial rectus. And again, it's straight. Up here, superior rectus. And down here, inferior rectus. And those are the four muscles that cows have, and humans too. And so we can move our eye this way, we can move our eye this way, we can move our eye this way, we can move our eye this way. But again, remember, humans have two additional muscles, and they run diagonally. Well, one of these, you can only see a little bitty corner of it right here, but it's on the bottom, and it runs diagonally, so it's called the inferior oblique. There's another muscle, too, that really you can't see on this model because the muscle's actually over here. So the muscle is like this, and then it has a tendon, and the tendon makes a curve and comes this way. Well, you can't see the muscle, but you can see the tendon. And so this is the superior oblique. So if we look at that, we see those muscles. So they're same here too, but what we have to do here is lift this off in order to see those muscles. And when we do, there they are. So if we lift this off, there's those same muscles that we're talking about. Here's our lacrimal gland, which means this side is lateral, which means this is the lateral rectus. There's going to be the medial rectus, the superior rectus, the inferior rectus, and so on. If we turn it sideways, you can see what those look like. So, any questions about any of those? I can't see the mouse, only the drawings. I don't know how to make that better. Yeah, I don't know really how to make it any better. Well, I'll just try to write more. How about that? Okay, so here's the medial rectus. On the other side would be the lateral rectus. Here's the top, that's the superior rectus. There's the inferior rectus. There's that tendon for the superior oblique, and right there is the inferior oblique. So, any questions about the muscles? Well, let's look at the lacrimal apparatus. So remember, the lacrimal apparatus is all about tears. Well, first of all, let's talk about why we even have tears. Well, the reason we have tears is because there are no blood vessels here. No blood vessels. If you have no blood vessels, that means that there's no blood across here. None of these tissues right here receive any blood at all. The reason we have no blood vessels is because we need the front of the eye to be clear so that light can get through there. If we had blood vessels running across here, they would block the light as it went through. So there aren't any blood vessels. Well, if you don't have any blood vessels, that means you don't have any blood. And if you don't have any blood, there's no nutrition. So we have to figure out some other way to supply nutrition to these tissues. And that's what tears are all about. So tears have oxygen in them, they have nutrients in them, and so on. They also remove carbon dioxide and waste products. And so what happens is we're going to secrete tears, and then when you blink, they're going to get wiped across the surface of the eye. So this happens all day long, all night long. Every time you blink, you're wiping tears across the surface of the eye. As the tears wipe across the surface of the eye, they're going to supply oxygen and nutrients. They're going to remove carbon dioxide and waste products. Well, those tears have to have some place to go. And they go into this part right here. And so they're little tiny openings right here. They're called puncta. So we have an upper punctum. That's that little hole right there. And we have a lower punctum. That's that little hole right there. And what's going to happen is the tears are going to drain into those little holes. And from there, they go into these little tiny tubes, little tiny canals. And the word for that is canaliculus, or plural is canaliculi. Kind of sounds like, can I lick your eye? But look, there's an upper canaliculus and a lower canaliculus. And then what's going to happen is going to go into this tube. This tube is called the lacrimal sac, and it's going to drain into your nose. So all day long, all night long, we're making tears. They're being wiped across the surface of the eye. They enter the little puncta, then the canaliculi, then the lacrimal sac, and they drain into your nose. And then the cilia in your nose move it to the back of your throat, and you swallow it. But think about what happens if you start crying. What happens if you cry? Well, when you cry, the amount of tears that comes out of here is much, much more than it is usually. And so some of the tears fall out of the eye this way. But a lot of those tears are going to go this way. They're going to go this way. What happens to your nose when you start crying? Anybody know? Your nose runs. Think about it. But it's not snot coming out. It's not mucus. It's tears that's coming out of your nose, these excess tears. And if you happen to taste that, if it were to run into your mouth, it would taste salty because tears are salty. Does that make sense to everyone? So if we look at that on the model, it looks like this. So here is the lacrimal gland right there. And that's what's going to produce those tears. It's going to go through these little ducts, these little green things that you see, onto the surface of the eye. When we blink, wipe across the surface of the eye. Right here's the puncta, right here and right here. And it's going to enter these canaliculi. Remember, there's a superior and an inferior canaliculus, and then here it goes into the lacrimal sac, and then finally down into your nose. So if we label those things, it looks like this. Well, we also know that we have eyelids. So the eyelids are there to protect the eye. They also act like windshield wipers to move the tears across the surface of the eye. But eyes are pretty delicate in the front. You don't want the things getting in there. And so we can blink our eyes, not only to move the tears, but to keep things from getting into the eye. Well, the eyelids are called palpebra. So we have an upper palpebra, upper eyelid, and a lower palpebra, lower eyelid. And what they're made out of is connective tissue covered in skin. So there's a connective tissue plate, if you will, that's covered in skin. And then, of course, we have eyelashes and so on. Well, if you look at where these eyelids come together, there's a little corner to your eye, a corner to your eye. The corner goes by two names. It's either called a canthus or a commissure. So that right there is a canthus or a commissure. That's another one. Well, again, they're named for the direction. Remember, this thing is always lateral. So this is the lateral canthus or lateral commissure. That makes this one the medial canthus or medial commissure. If you look at medial commissure, you're going to see a red spot. If you look in the corner of your own eye, look in the mirror, you're going to see this red spot. That is called the lacrimal caruncle. And what you find here are glands that are very similar to sebaceous glands. And so they're producing this oily secretion. The oily secretion is there to get spread across the surface of the eyelid. It also helps to keep the eyelashes flexible and less likely to bend or break, fall out. These glands that are in here secrete constantly, even at night. So at night, when you close your eyes, they keep secreting, but there's nowhere for it to go because your eyelid is not opening and closing. It's not moving it. And so at night, these secretions tend to collect here. And so when you wake up in the morning, these secretions have collected here and they've dried up. So when you wake up in the morning, you have this sort of crusty-like stuff right here in the corner of your eye. The eyelids are attached to the eyeball, and they're attached by what's called a conjunctiva. Conjunctiva is a membrane that attaches the eyelid to the eyeball. So if your eyelid was here, there's a conjunctiva that does this. It covers the inside of your eyelid, and then it comes down and attaches to your eyeball, like in a circle, like this, all the way around. And so there's a membrane then that lines the eyeball or lines the eyelid and attaches to the eyeball. So if you wear contacts or something like that, people think they lose their contact in their eye and it gets down around behind their eye. Well, that's impossible because there's this membrane that prevents anything from getting behind the eye. And here's that conjunctiva right here. So you can get an infection of the conjunctiva, an inflammation, and you get what's called pink eye or conjunctivitis. Any questions so far? So let me go back and answer this question. And that is about the muscles. So remember when you talk about muscles, muscles can really only do one thing. They can only pull. So if this muscle pulls, it's going to pull the eye this way. If this muscle pulls, it's going to pull the eye the other way. My cursor's stuck for some reason. Can everybody still hear me? Oh, I don't know what's going on. My cursor's stuck. I may have to leave the session and come back. So just hang on and I'll try to do that. Although I don't know how I'm going to do it. Looks like my mouse became disconnected to the computer. But anyway, we'll use this mouse. So again, if this muscle contracts, it's going to pull the eyeball. It's going to contract and pull like this, and the eyeball is going to go this way. Same thing for this, and so on. And so whenever you talk muscles contracting, again, they can only pull. So her question was, do rectus muscles allow movement to sides, up and down, and oblique diagonal movement? Yes, exactly right. So if I only contract this muscle, remember this is lateral over here. So here's my nose over here. So if I contract this muscle, the eyeball is going to look toward the nose. If I contract this muscle, the eyeball is going to look toward the side, toward the temple. And then because we have these diagonally, I can also look up, but not straight up off to the side and down to the left, and so on. Okay, so that's the outside of the eyeball. Let's look at the inside of the eyeball. So if we cut open the eyeball, it's hollow in the middle. And so if you look here, you can see a big hollow space. Well, it's not empty, it has fluids in it. And there's actually more than one hollow space. So if we look here, there's a dividing line, and here's the dividing line. This structure right here is called the lens, and the lens divides the eyeball into two what are called compartments. So there's an anterior compartment. Remember, anterior means toward the front, and there's a posterior compartment, which means toward the back. And so there's a hollow space right here on this side of the eyeball, like this. And then there's a hollow space right here on the back side of the eyeball. Well, again, they're not empty. They have fluids in them. And the fluid that's in this front compartment, this anterior compartment, is called aqueous humor. Aqueous humor. So if you look at aqueous humor, aqueous means watery. So it's very watery. Now, it's not water, but it's watery. It's very, very liquid. But if you look at this one back here, it has something in it called vitreous humor. And vitreous means thick. And so this liquid is like jelly. It's thick, like jelly. Well, we constantly make and we constantly drain off aqueous humor. We're making it all the time. This one you don't make all the time. You make it when you're a fetus and a small infant, and then it stays with you the rest of your life. It really doesn't change very much. So whatever you finished making after you left infancy, that's what you have right now. So as you get old, it gets old. Well, light passes through here and all the way through to way back here in order for you to see. And so remember, we won't want to block out light, so we would want this liquid to be as clear as possible. But remember, this liquid gets old. As you get old, it gets old. And what happens to it is the same thing that might happen to, say, jelly if you left it in the refrigerator for 20 or 30 years. It begins to become discolored. It turns sort of a yellow color. And so old people have a yellowish tinge to their vision because of this vitreous humor. So there's our aqueous humor. There's our vitreous humor. Any questions about either of those? Let's look at the aqueous humor first. So if you look at this front compartment, the anterior compartment, remember it's everything in front of the lens. But look, there are two spaces here because this right here is a hole. That hole is the pupil. It's the pupil. And it's a hole, literally a hole, which means fluid can move back and forth between these two spaces. But these two spaces are not the same space. They're connected, but there are two chambers. So it has two chambers, an anterior chamber, so that's in front of the pupil. And it has a posterior chamber, which is behind the pupil. But remember, there's a hole connecting those two, and so fluid can move back and forth, back and forth. So this fluid that moves back and forth, is very, very similar to tears. Remember the reason we have tears anyway? It was to nourish the front part of the eye out here because there are no blood vessels. Well, look, there are no blood vessels in here either. And so we also have to nourish this part of the eye as well. And that's what this aqueous humor does. It nourishes the front, these two chambers. So this thing that's hanging down the pupil, the front of the lens, all of that. And we're constantly making it, and we're constantly draining it off. So it's made by this structure right here, this black structure you see. We'll talk about it more in a minute. But it's made, and it's going to go through here. It's going to circulate. And then see this little vein right here? Now it goes all the way around. There's not just one of them, but that's where it exits. So there's constantly new aqueous humor being made. It circulates, nourishes, picks up waste products, and then it exits. It's a one-way circulation. Does it make sense to everyone? So here's that structure that makes the aqueous humor. Again, we're going to talk about it more in a minute, but it's called the ciliary body. And then here's where the fluid goes. It goes in here, and it nourishes the front of the lens. It nourishes the back of this tissue here. And then it goes through the pupil. And then it nourishes back of the clear part of the front of the eye. And then eventually it's drained off in these little holes, which drain into the veins. So again, it's a one-way circulation. Any questions? This picture shows the same thing. So here's our ciliary body. That's what makes it. And there you see the circulation. And there's the little exit right there. Well, let's look at this ciliary body. Not only does a ciliary body make aqueous humor, but it has another job too. Not only does it have this secreting portion, but it also has a muscle. So there's a muscle that goes all the way around the front of the eye like this. It's the ciliary muscle. And in the center of that ciliary muscle, suspended is the lens. So here's the lens like this, and it has little tiny ligaments. They're called suspensory ligaments. So here are the suspensory ligaments, and you can see them here. That's what these white things are here. and here. And they attach the lens to this muscle. Attach the lens to the muscle. So the muscle surrounds the lens. It's a circular muscle. So it looks something like this. This other mouse is harder to draw with. But anyway, think about if it's a circular muscle. When a muscle contracts, what happens is the inside diameter gets smaller. When a muscle relaxes, the inside diameter gets bigger. So remember, the lens is suspended inside this muscle. So when the muscle relaxes, the tension on the lens goes down. There's less tension, less pulling, less stretching. But when this relaxes, because it's going to open up wider, the tension on the lens is going to go up. Well, the lens is soft. It's soft. And so if I take and apply tension to something that's soft, it's going to stretch. And then if I let the tension go, not only is it soft, but it's elastic. Then it's going to go back to whatever shape it was. And so what happens with this muscle is it changes the shape of the lens. So if the muscle contracts, so here's my lens. Lens is kind of a sphere like this, but remember it's soft and elastic. But look, if I pull on it this way and pull on it this way, when that muscle relaxes, it's going to be shaped more like this. It's going to be shaped like a pancake because it's been stretched. But if I let this relax, what happens is that pancake shape, when I let it relax, it's going to go back to this round shape. So I can change the shape of the lens. It can be either big and round, spherical, or it can be pancake shaped. And it's this muscle that does that. So here's the muscle. There's those little tiny ligaments. And then here's our lens. So again, think about this. If this muscle relaxes, it's going to go this way. It's going to relax out like this. So look, the tension on here is going to be greater, and it's going to stretch the lens. So the lens is going to look like a pancake. But if I relax, that's if I relax the muscle. But if I contract the muscle, remember the muscle is going to go inward. It's going to go here and here. And look, there's going to be less pulling, less tension on these suspensory ligaments. And so the lens is going to fatten up and it's going to be more spherical shaped. That kind of looks like this. So look, there's the lens, there's our little suspensory ligaments, there's the lens, there's the little suspensory ligaments. Here's the muscle. So when the muscle contracts, this is what happens. Here it contracted. And look at the distance between here and here. It's shorter. So look, these ligaments have less tension. And when they have less tension, look what happens. You get this spherical lens. But here, this thing relaxed. Look at the distance here now. It's this far. And what's going to happen is it's going to pull backwards. I'm going to increase the tension. And when you increase the tension, the lens is going to flatten. It'll look like a pancake. Does it make sense? Well, the lens is a little bit like the kind of thing that you might have for a microscope or a hand lens or something. So if you think about like a magnifying glass, you probably have had one of these when you were a kid. It might have had a handle on it and you went around looking and things and stuff like that. Well, if you look at this lens on a magnifying glass, it's shaped like this. It's shaped like that. And the thicker this middle part is, the more it magnifies. So look, the lens is just like that. And the thicker this middle part is, the more it magnifies. And that's what happens when we look at things either close up or far away. When we look at something very, very close, like the words that you might read that say fish on a piece of paper close to your eyes, this thing has to be fat because that bends the light and makes it go here. But when something's further away, I need to bend the light less. And so I don't need this to be as fat to make the light here. And when something's very far away, I don't need it fat at all to make it line up. So remember, in order to get this to happen, what has to happen here is the muscle has to contract. We're going to relieve the tension on the lens, and the lens is going to get fat. But here, look, in order for this to work, the muscle has to relax. It's going to get wider. And when the muscle gets wider, we're going to increase the tension, and this thing is going to turn into like a pancake. Just like this. So this is for close vision. And this is for far vision. So this is for close vision. And this is for far vision. So let me ask you a question. Close vision or far vision? Which one requires the ciliary muscle to contract? Close vision or far vision? Close, exactly. So when you contract a muscle, after a while, muscles get tired. So have you ever noticed that if you are reading for a long period of time or sewing or doing something very close up, your muscles get tired? I mean, your eyes get tired. But when you're looking at something far away, these muscles are relaxed, and relaxed muscles don't get tired. And so when you're looking at something far away, you don't feel that way. They don't get tired. I really hate this mouse. Does that make sense to everyone? Any questions? Okay, so there's our lens, there's our ciliary muscle, there's our little suspensory ligaments, and then there's the ciliary body that makes those aqueous humor. Well, let's look at the eye some more. The eye has 3 layers to it, and a layer is called a tunic. The word tunic means coat. So it has 3 layers or 3 coats. There's an outer layer, which is called the fibrous tunic. Fibrous tunic, made out of fibers, thick and tough. There's a fibrous tunic. There's a middle layer, which is this black that you see in here, plus all of these blood vessels that you see. It's called the vascular tunic. Vascular blood vessels, got lots and lots of arteries and veins running through here. Now remember, there are none over here, but there are lots of them in the back of the eye. I don't know what that is. So that's the vascular tunic, this middle layer. And then there's an inside layer. And the inside layer is actually clear. It's transparent. So if you look right here, you can see this little thin milky colored layer that's right on the surface. Well, that covers all of this, but it's clear. So look, I can see blood vessels through it because it's clear, but it's made out of nerve cells. It's made out of neurons, and so it's called the sensory tunic. So 3 layers: fibrous, vascular, sensory. Well, let's look at that again. There's the sensory tunic right there. It's clear, so we can see blood vessels through it. But remember, it's made out of neurons. But let's look at the fibrous tunic first. So again, fibrous means fibers. It's thick, it's tough. So this is the part of your eyeball that holds everything in place, the part of your eyeball that gives its shape to it. But it's also the front of the eyeball, which is clear. So when we look at it, there's 2 parts to this. The white of your eye is called the sclera. And sclera comes from the same root word as sclerose, which means to scar. So it's made out of the same kind of tissue that scars are made out of. It's dense, irregular connective tissue, if you remember that from AMP1. Has a ton of collagen. And that collagen runs in all directions. So it's opaque, it's clear, or it's not clear. It's opaque, you can't see through it, and it's tough. It's what the muscles are attached to. But this is it. It goes all the way around, except in the very, very front. All the way around, except in the very, very front. It's the white part of your eye. But in the front of your eye, we also have fibrous connective tissue. This part, though, is clear. And the reason it's clear is because the collagen fibers are parallel to each other like this. And they're thin. I'm not drawing it very well, but they're thin fibers. They're like this. And look, light can pass through there because there's a space between the fibers. The light can pass through. Think about yourself if you're looking at, say, I don't know, you go to the zoo or something and you look at a cage. Here's the cage, but there's an animal behind the cage. And so you focus on the animal back here. Maybe it's a monkey or something. You focus on that, and these bars seem to disappear. And what happens is your brain erases them. That's exactly what happens here. Your brain erases these fibers. So when you look, you don't see them. The light just passes through between the fibers, but they're still there. So the front of the eye, this cornea, is actually quite tough because it has a lot of collagen. Now, it's not as tough as this, but it's still very tough. Look how thick it is, too. It's quite thick. So if any of you have ever had LASIK, when you have LASIK, what they do is they zap the front of your eye with a laser, and they reshape it. So they change the shape of the cornea. So instead of it looking like this, it might look like this. Well, maybe not like that. But anyway, and so when the light comes through here, it gets bent. Anyway, and that can help improve your vision. But there's our sclera. That's the white part of the eye. See, the muscles are attached to it. It's the strongest part of the eye. There's the cornea. It's the clear part of the eye. And again, look how thick it is. It's quite thick. There's the cornea right there. There's the sclera over there. Same thing here. Okay, let's look at this middle layer. So the middle layer is called the vascular tunic. Vascular refers to blood vessels, and it has lots of blood vessels, lots of them. So you can see arteries and veins in here and so on. Well, it's also got some other parts to it. One part we've already talked about, and that's the ciliary body. So here's the ciliary body. And remember, it has part ciliary muscle, and it has these ciliary processes which make the aqueous humor. So when we look at the ciliary body, it's in the front of the eye, and it surrounds the lens. And remember, the ciliary muscle is attached to the lens by these little suspensory ligaments. Another part that we haven't talked about is the iris of the eye. So the iris is the colored part of your eye. So if you have brown eyes or blue eyes or green eyes, that's the iris. And the iris is also part of this vascular tunic. And in the center of the iris is where the pupil is. And then the last part of this is called the choroid. So the choroid is the blood vessels. It's the vessels. It also has a pigmented layer, which is dark. So the back of your eye is dark. It's pigmented. Again, I don't know what that is. But anyway, there's that ciliary body. There's the iris right there, the colored part of the eye. And there's the pupil. Remember, it's a hole. It's going to let the light go through. Same thing here. So here's the choroid. Here's the iris. There's the pupil. So if you look in a real eye, it looks like this. There's that white part of the eye. Remember, that's the fibrous tunic. You can't really see it, but covering this right here like a dome is the cornea. And then if you look through the cornea, remember it's transparent. You can see the iris. That's the colored part of the eye. And then that hole in the middle is the pupil. Looks like this. So choroid, all those blood vessels, also pigmented. There's the iris, the colored part of the eye. There's the pupil. Well, the reason we have an iris is not to make your eyes pretty, even though it does. It controls the size of this hole. So the pupil, we can open it up. We can dilate it. And that lets more light in. So if you're in a low light situation, the pupils dilate so that more light can go in. But we can also close it up. We can constrict it. And that's to keep the light out. So if you're in a very, very bright situation, too much light, you can't see anything. And so we close it up. Well, it's the job of the iris to do that. The iris has two muscles that do it. There's a radial muscle, which dilates it. And then there's a circular muscle, which constricts. So two muscles. And look at the difference in the pupil sizes that you can get. But here is the radial muscle. Again, that's going to dilate it. It actually physically pulls the pupil open like this. It pulls in this direction, this direction, this direction. But then there's also this circular muscle right here. And when the circular muscle contracts, this is what happens. The internal diameter gets smaller, just like we talked about when we talked about the ciliary muscle. Well, both of these are controlled by the autonomic nervous system. And if you remember, the autonomic nervous system has two divisions. It has a parasympathetic and a sympathetic. Parasympathetic causes constriction. So we wind up with a small pupil. Sympathetic causes dilation. So we wind up with a large pupil. Any questions about any of that? So there's those two muscles. There's a picture of the choroid that we can see very nicely. Remember, the choroid is pigmented, but it's also got a lot of blood vessels. But it's pigmented, it's colored. And then remember, in there we have the vitreous humor, and there's the lens. Emmy, the quizzes will show up at 3 p.m. after we do the collaborates every time. So if we open it up, there's our choroid, there's our ciliary body with the ciliary muscle and so on. And then here's the back of this iris. There it is right there, and there's the pupil. But there's the ciliary body behind that. Okay, let's look at the sensory tunic. So remember, the sensory tunic is... Is anybody having any trouble hearing? I don't know what to do. How about now? Is that better? Okay, well, let's look at the sensory tunic. So remember, the sensory tunic covers this whole thing. So all of this is the sensory tunic. But remember, it's clear. So we can see through it, and we can see blood vessels and all that. But it covers this whole thing. It's a very, very, very thin layer. It's not really attached to anything, but it gets pushed against these other layers by the vitreous humor. So one of the jobs of this vitreous humor is to hold this sensory tunic in place. If you get hit on the head, especially the side of your eye or something, you can dislodge this sensory tunic. In other words, instead of it being nice and flat, it'll fold up right here. And so if light hits that folded area, it's going to be distorted. You're going to have distorted vision. But let's look and see what that is. Well, what they did for you is they pulled out over here a blow up. So this is a blow up the sensory tunic. The sensory tunic is called the retina. And this is a blow up of it. And look, it's nerve cells is what it is. Here's what it really looks like. So look, it's sort of this, and this is a fixed one, which means that it's been put in fixative to keep it from rotting and stuff. But what that does is it makes this clear retina become cloudy looking. But it also makes it easier for us to see it. So that's that. And then here's where the vitreous humor is. And remember, it's going to push against that and hold it against the vascular tunic. Now we took the vitreous humor out of here, and so look what's happened. It's detaching. It's not supposed to have these ridges in it or anything like that. It's supposed to be smooth, almost as smooth as glass, because light is going to hit this, and this is where the cells are that are going to detect the light. Well, if it's folded up like this, light is not going to hit it at the same angle that it would as if it were flat. Well, when you look at it, this is where those nerve cells are. It's where our photoreceptors are. We'll come back to this in just a second. But here are the photoreceptors right here. And so if you look at photoreceptors, they come in two types. There are these, which are called rods. That's just because that's what they're shaped like. And then there are these, which are called cones. Again, it's just what they're shaped like. These detect light. But they're not the only kind of nerve cells that are in here. These are also nerve cells. And these are nerve cells. And these are nerve cells. Well, light actually passes this way to get to these photoreceptors. It has to go through here to get to here. Go through here to get to here. And so as it goes through, a little bit of the light is blocked out. Well, that seems like it defeats the purpose, and it kind of does. But again, we have two types of these photoreceptors. We have rods. Well, rods see in low light, and there's only one kind of rod. And so what we're going to get are shades of the exact same color. There's shades of gray, because there's only one type of rod. There's only one color. But cones, when you look at them, they're for high light, lots of light, and there's three types of these. Look, they have them painted. They're not really these colors, but these are the colors of light they detect. So look, that one detects the red portion of light. That one detects the green portion of light. That one detects the blue portion of light. And so because we have three different cones, we can see combinations of these different colors. And so depending on what kind of color you're looking at, you stimulate them more or less. And so you can see thousands of colors, not just one color with different shades, but thousands of different colors. Well, let's go back to our other picture. Rods are pretty much everywhere, except there are no rods right here. But there's rods out here, just pretty much everywhere you go. cones are also pretty much everywhere, but cones are concentrated right here. So that's where they have tons of cones. And then there's another place, and that's right here, which is where all those nerve cells are going to exit the eyeball, and there are no rods and no cones, which means there's no photoreceptors, which means this is a blind spot. So all of our eyes have a blind spot because when light hits that, there's no photoreceptors there. And whatever that is, whatever it's coming off of, you don't see it. But what happens is our brain fills in the blank. So we don't, when we look at things, we don't see a gap. Our brains fill in the blank. But again, rods basically everywhere. So here's the rods, except here, no rods. Cones, basically everywhere. And they have the highest concentration right here. That's where they are in high number. And then this spot where there's no rods and no cones, none, neither. And so it's a blind spot. So let's look at the names of these places. So the rest of this is just called the retina, but this has a name, and it has two parts to it. Around the outside edges is called the macula lutea. Macula lutea is, macula means big and lutea means yellow. It's a big yellow spot. And there it is right there. That's the macula lutea right there. Look how different it looks than the rest of this person's eye. And then right in the very, very center of the macula lutea is a sort of pit. And the word for pit is fovea. So it's called the fovea centralis because it's right in the middle of the macula lutea. So right there is where the fovea would be. Right there is where the fovea would be. So remember, we have these two places. This is where all of those cones are. Cone, 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 cone. But cones, remember, attached to another cell. And they're attached to another cell. And remember, the light has to go this way to get to those cones. And so some of this light would be blocked off. But what happens is, in the fovea, it's shaped like this because these other cells are folded off to the side. And so the cones are exposed without any kind of blocking, which means that the light that comes to the fovea is not blocked out, it's not obscured, and so it's going to have the clearest picture, the clearest vision. And so this fovea and this macula are where we have the highest number of cones. And this fovea, remember, there's nothing blocking it, so we have the highest visual acuity. It's where your absolute best, clearest vision is. Because all of these cells are folded off to the side. So here's our rod. Rod, remember, shades of gray. And they work in low light. So if you walk outside right now and the sun is shining bright, 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 these are going to get bleached out really quickly and they quit working. They get bleached out. So you ever notice what happens when you go back inside and it's dark? For a little while, you can't see anything. The reason why is because these don't work in low light. They only work in bright light. And you've been outside, and so these have been bleached out, so they're not working temporarily. But if you go outside at night, these don't work. And so if you've ever noticed when you go outside at night, you don't see color. Even if you look at, say, like a rose, that rose is not red. It's some sort of shade of gray because there's just not enough light. You need bright light. They only work in bright light. Here's what they really look like. Can everybody tell which ones are rods and which ones are cones? So what are these right here? Yeah, and what are these? So you can see the difference. It's not hard to see. And then here's those other cells that are also important. Well, if we look over here, remember this is a blow up of this. See how these are folded off to the side right here? Well, that's what happens in this fovea. And so the light can just go straight to those photoreceptors without having to go all the way through here to get to the photoreceptors. But when we look here, we can see rods. So these are rods. And we can see cones. Those are cones. They connect to another type of nerve cell, these, which are called bipolar cells. So if you remember from A&P1, we talked about neurons. Remember they were categorized by shape. We had unipolar, bipolar, and multipolar neurons. Bipolar are only found in the special senses. So they're only found in the eye and the ear and nose and mouth. That's it. But there they are right there. Those are the bipolar cells. And then they connect to another cell. These are called ganglion cells. So these are ganglion cells. And all of the ganglion cells, their axons become the optic nerve. And so it's going to carry it to your brain. We also have some helper cells in here, little weird cells, which are called amacrine or horizontal. So we go back to this picture here. Here's the amacrine cells. And here's the horizontal cells. But there's our rods and our cones. Here's our bipolar cells. Here's our ganglion cells. And then we have these amacrine and horizontal cells. The only cells in this whole group that can detect light are the rods and the cones. The rest of them are just to pass the action potentials along to the brain. Well, look at this. Here's our rod. Remember, rods only detect dim light. So if the sun is shining, look what happens to the rods. They sort of go to sleep. They're bleached out. They don't work at all. But what does work are the three cones. So we have red cones, blue cones, and green cones. Well, look, these guys are holding hands. The reason they're holding hands is because there's a lot of overlap between the light that they detect. Whereas this guy is not holding hands because the light that it detects is separate. I really hate this mouse, but anyway. So if we look at that, here's what that looks like. Look, there's a lot of overlap here, red and green. There's not much overlap with blue. Well, what would happen if your cones are not working? Well, if your cones are not working, you can't see a particular color. And so it depends on which cones are not working as to what the colors are that you see. And so different diseases affect different types of cones. And so look, this is called protanopia. And protanopia, there's only one cone that's messed up. So one cone is not functional, or it's not functional completely. And so when you have this one cone messed up, what happens is you confuse colors. You can also, though, have two cones messed up. So if you have two cones, you're going to see even more messed up colors. And so I can't do this on here, but if you do the PowerPoints and you click on this, it'll take you to a website where you can see these individual, what happens with these individual colorblind or color deficiencies. And so it'll have a picture for you to look at, and then you can click and see what happens if you're a proteranope or if you're a deuteranope, and see what the person would look like. I wish it would work on this, but it doesn't work on this. But you can certainly do it. And then finally, remember the eye, even though it detects light, this is not where we perceive what we're looking at all. It's back here in the brain. And so somehow that information has to be sent all the way back here. So remember, we started with all these different cells. And so we started with the cones and the rods, and then we're going to create action potentials. And so we're going to get action potentials. And then the action potential here, we're going to get synaptic transmission to these cells, and then synaptic transmission to these, and so on. Well, if you look at this right here, remember when we talked about summation back in AMP1? Look at this. All three of these rods are attached to this one cell here, which means that light hitting this one is going to wind up on this cell. Light hitting this one is going to hit up in this cell. When this gets to your brain, you're not going to be able to tell the difference between this and this and this and this. In other words, this is going to have sort of a fuzziness to it. It's not going to be very sharp. But because there's three of them on one cell, it can collect a lot of light. And so you can see in low light places, because it collects a lot of light, 3 times as much light. But if we look at cones, look, cones don't do that. So if we look at this cone, it's basically a pretty straight thing all the way through. And so if you look at this one, look, it's straight all the way through. And so we don't get fuzzy vision, we get crisp vision. But because it's a 1-to-1 ratio, you got to have a lot of light for it to work. You have to have a lot of light. Otherwise, you don't see anything at all. But we're going to pass that information. All of those axons of those ganglion cells are going to form this optic nerve. So this optic nerve comes from this eye, this optic nerve comes from this eye. And then when you did the brain, you should remember the optic nerves, which is cranial nerve number 2, is going to cross right here, and we're going to form the optic chiasma. And then they're going to separate, and we're going to form the optic tracts. And where they do then is they go to the thalamus. And then from the thalamus, they get passed along to the primary visual area. One last thing, and we'll be done. Look at these fibers. Some fibers cross. Look, they go to the other side. But some fibers stay on this side. Which means that both sides of your brain get a complete picture. Because it's half here and half here, half here and half here. Both sides of your brain get a complete picture. If it weren't like that, if this whole eye crossed over to here and this whole eye crossed over to here, each side of your brain would only get a half of the picture. But it's not like that. Does it make sense? So that is basically the eye. So does anybody have any questions? So remember around 3 p.m. and you'll get a notice in your email, and there'll also be an announcement, but the quiz will become available. And so if you want to do the quiz today, you can. And I think that you should, but you don't have to. Any questions? Also, don't forget that if you want to go back, you can look at the recording of this. I'll try to get my mouse fixed before next time. How can some animals see in the dark? Well, we all see in the dark to some extent. But remember, the cells that you use to see in the dark are rods, right? So what if I only have 2 rods compared to I have, I don't know, 100 rods? Do you think I'm going to be able to see equally with 2 rods versus 100 rods? No. So animals that see very, very well in the dark have a ton of rods. Now, they have some other adaptations too. They have bigger pupils. They also have a layer in the back of their eye which reflects light. It's called the tapetum. But humans don't have a tapetum. So how to intuitively understand the lens changing okay. It's a little hard, and I'm having a hard time drawing with this mouse. But the thing you have to remember is the lens is suspended inside like a ring. So it's stretched. So it's stretched by this ring. But let's pretend this ring is flexible, and I'm going to take my hand and I'm going to push the ring like this and push it like this. So what's going to happen is this is going to be, when I push in, the lens is going to be less stretched. And if it's less stretched, it's going to get fat. So it's going to go from a stretched shape, which might be like this, I really cannot draw with this mouse. It's going to be less stretched, so it's going to get fatter. So that's what happens with this. It has to do with the muscle being in a circle. So remember, a circular muscle, when it contracts, the inside diameter gets smaller. When it relaxes, the inside diameter gets bigger. And then the lens is suspended inside it. So look, if this is my lens right here, right now those little suspensory ligaments are straight because they're being stretched. And the lens is being stretched. So if I look at this lens from the side, here's my suspensory, really hate this. Here's my suspensory ligament. And here's the lens like this. It's going to be shaped like this because it's being stretched. But now if I take that same size lens, I don't know if I can draw it in here, but there's the same size lens. Look, these little ligaments are now loose. They're not bent. There's no tension on them because it's so close. So if the ligament is loose, there's no tension on it. I'm not going to be pulling on this lens. The lens is going to get fatter. It's going to go this way. So again, when you contract the muscle, the lens is going to get fatter. If you relax the muscle, the lens is going to get thinner. So to see close up, we contract the muscle. The lens gets fat. To see far away, we relax the muscle and the lens gets thin. Does that make sense? So she says, is it like a rubber band stretching? Well, these things are like a rubber band stretching, I suppose, yeah. But the hard part for most people is understanding that the muscle has to contract to relax the lens. And when the muscle relaxes, it stretches the lens. Wow, this is an awful mouse. I promise to get that fixed very soon. Okay, well, I'm going to go ahead and stop recording.